The wee green things in the corner that says we're live. It's been telling the truth for weeks. Is it telling the truth tonight? We'll soon find out. But I won't be doing a JD Kane because I've muted the actual playback. I did that ahead of time. Yay,
Brilliant.
me! Yeah, I know. Anyway, this week, it's the usual science, bitches. As in, me and Jade. And we have Mel here as well. And we have the honorary bitches, RSC, known as RC to his friends, and Cliffjumper. The link has also got to Hog Tie and Ian if they're able to make it, and I hope they can. Anyway, this week, we were going to pass out some information about a virus that we've all heard of, and we all know a little bit about. Most people have heard something about it, but they don't have the sort of the full picture of what is it about this virus that's such a horrible thing. So, I don't know how many of you out there are old enough to remember when AIDS first appeared. And it first appeared in San Francisco, and I remember following it. And nobody knew what caused it, and there was all sorts going on. And they were identifying this high incidence of Kaposi sarcoma in these patients that all showed similar symptoms, and nobody knew what was causing it. And I remember the day that I was in work in a diagnostic bacteriology lab with the radio on, and it came across the radio that they had identified the virus. And at that time, they called it HTLV3. And I still remember hearing that where I was standing the day that I heard the news. It's been a virus that most of us have grown up hearing about, but we don't have a lot of the details. And a lot of myths have grown up around it. There's a lot of stigma has grown up around it. And so we decided to put out some information, maybe bust some of the myths, and hopefully the stigma has already been bust, but we'll just deal with that as well. So I'm going to hand this over now to Cliffjumper, who's going to go through a description of the rise of the virus, where it came from, how it got to where it went, and how it got to where it is. So I shall now mute myself and put my camera off. And Cliffjumper, you can take over, dear.
Thank you very much. You just want to present me or give me the powers to present myself? Thank you very much. So just going to share a picture of the world here. Flat earthers. Oh, they'll go crazy. Anyway, for most people, HIV and AIDS is something that came into the public consciousness in the early 80s. And most books and websites about AIDS started in 1981, where when gay American men started to die from a rare form of pneumonia. However, the HIV virus goes back almost a century. And unlike what the religious zealots might say about AIDS was caused by the gays, gay people are at the end of the chain, not the beginning. In fact, HIV/AIDS has its roots in the Congo region of Africa, Central Africa, so this part of the world here. Before I go into the virus itself, it's important to give a short overview of the history of the Congo. In the late 1800s and into the early 20th century, there was a scramble for Africa, with several European countries occupied, divided, and colonized almost all of Africa, right down to having a multinational agreement on how it would be divided up and trade regulated without European powers descending into expensive wars over territorial claims. In 1870, only 10% of Africa was under European control. By 1914, over almost 90% of the continent had been taken. Along the Congo River, the Belgians and the French took territory on either side. The French took over an area of the north side of the river in 1880 as a colony known as the French Congo. So this is this part here. And on the south side of the river, it pales in comparison with the administration of King Leopold II of Belgium, who held the region as his own private property. And it was, this is what is now the Democratic Republic of Congo. And it was ironically called the Congo Free State. Brutal exploitation of the indigenous population forced them into producing rubber, to the point of making it a matter of policy of cutting off the limbs of the natives if they didn't meet the rubber quotas. Bound to international pressure, the Belgian parliament voted to annex the area in 1908 and re-establish it as a Belgian colony. The HIV story begins around 1921 between the Sanaga River in Cameroon, which is up here, and the Congo River, where a minority species of chimpanzees were contained due to the chimps being unable to swim and less able to spread. In this region, the bushmeat trade was a vital part of life, the meat coming from chimpanzee, gorilla, monkey, rat, deer, and even fruit bat. The ancestor of HIV comes from a subspecies of chimp called the pan-troglodytes, that wasn't a stutter, who, like us, are omnivores who hunt and eat red-capped mangabeys and mustached guenons, who are natural carriers of a simian immunodeficiency virus, SIM, with around 6% of troglodyte chimps ever infected. Within a troop, each female mates with many males, but mating with outsiders is rare, so most troops were untouched, where a few are heavily infected. As you know, HIV is best transmitted via the blood. Chimps are our closest relatives, sharing between... sorry, am I not presenting to everybody? There you go. Sharing between 98 and 99% of our genome with us. So thanks to the bushmeat trade, the virus jumped from chimpanzees to humans. This probably occurred due to being scratched or a hunter cutting themselves during the butchering process while handing bloody meat, so blood-to-blood contact. Mel's going to talk about genetics, but HIV has 4 genetic groups, M, N, O, and P, showing that it made the chimp-human species jump at least 4 times in the past, but the M group accounts to 99% of all cases. The focal point of the virus emerged around the twin cities of Kinshasa and Brazzaville. That's here, and you've got the Congo River that goes between them. And they face each other. Colonialization led to these cities expanding rapidly, bringing trade and opportunity. And of course, like everywhere, prostitution came with it. However, sexual transmission isn't very effective at passing on HIV. Married couples can have intercourse for many months with it before passing it to their partners. And even though there was a history of polygamy in the region, the virus spread much quicker than sexual transmission would allow. So there must have been an amplifier. Another side effect, another side effect of rapid increase in cities, disease is also able to propagate. So the 2 colonial powers began to vaccinate the population. As I mentioned before, blood-to-blood contact is the most effective way to cross-contaminate people. And until the dangers of blood contamination were properly realized, syringes were used many times. A dose sucked into a syringe, used, refill from the same vial, spreading the disease rapidly. But how did the HIV virus get to America? Well, at the end of colonialization in the 1960s, it led to poverty, disease, and famine. Without an African lead, because the Belgians never trained one, there was no one to lead the nation, so the United Nations hired bureaucrats and teachers from abroad. One of the nationalities that stepped in were about 4,500 Haitians. They were black, they spoke French, and they could earn much more money than they could at home. In their time off, many Haitians would use the brothels and receive vaccinations, so when they went home to Haiti, they took the infection with them. The capital of Haiti is Port-au-Prince, and in 1971 and 72, there was a plasma center called the Hemo-Caribbean, which was known to have low hygiene standards. It was co-opted by Luca Cambrione, I think that's how you pronounce it, the leader of the secret police. Nicknamed the Vampire of the Caribbean, he He bled 6,000 sellers who were paid as little as $3 a day and exported 1,600 gallons of plasma to the United States each month. The first Americans to acquire HIV were, in fact, hemophiliacs who needed blood factor VIII, which is derived from blood plasma. HIV was also a prime destination for gay American sex tourists. The Spartacus travel guides even describing how much young men expected to be paid for, for sex. Young gay guys could go there, and their dollars would buy more, and they'd have a good time, and they'd catch us, they'd get laid, and they'd catch a tan as well. Jay's more familiar with this part, so I'll finish at her to finish off the story. But the modern history of AIDS had begun. Go for it, Jade.
Okay, thank you. And your timing is excellent because I was eating. So just give me a second. It always works that way, doesn't it?
Put your teeth in, Jade.
Yep. So AIDS entered the United States. Now, there was a myth about patient zero for a long time. It was supposedly a male flight attendant. And this poor guy was put through hell for years because everyone thought he caused it. Well, as Cliff pointed out, that's not true. That was a misunderstanding. So AIDS first entered the gay male population here in the United States via the path that the Cliff Jumpers already mentioned. And it became fairly common, unfortunately, in places like San Francisco, which was a gay mecca at the time. Because although San Francisco wasn't the friendliest place for homosexuals in the world, it was a lot better than a lot of other places. And there was already a very well-established gay community. Now, one of the interesting things about this, and it's very sad as far as the gay community goes, but it's very good for the rest of the United States, is the fact that HIV did enter this country in the gay community. Because the gay community was a small community, a small population of people, and for the most part concentrated in certain areas. As I said, gay communities like San Francisco. The fact that HIV did enter this. You're going to have to mute your feed there, Evie. Got it. Good to see you, by the way. So because it was in this small community, they were able to identify it. But HIV is the virus. AIDS is the syndrome. It's called acquired immunodeficiency syndrome. So you can have the virus and not develop AIDS. What was happening in San Francisco is that physicians and other medical personnel who were, who attended the gay community, because a lot of gay people went to doctors who were friendly because a lot of doctors weren't friendly. I could tell you stories about that one. And they started to see these rare disorders, like Kaposi sarcoma, which is a form of cancer, and that has a viral association. But it's very rare. Why was this thing short? Also, toxoplasmosis. Most of us have been, toxoplasmosis is caused by a protist. And most of us have been exposed to it because it's carried by cats. And even in the wild, even in like places like India, or even where people don't keep pet cats, you're probably still going to have been introduced to it because there are wild cats in the area. So the particular protist, most of us have had some association with it. But to become ill from toxoplasmosis, to develop, like a very bad case of toxoplasmosis, is very rare, except in like small children or babies, which is why pregnant women are told not to clean out cat boxes. Because even a small child, like a 4 or 5-year-old, if they get infected with this protist, they're probably going to be able to fight it off. But a developing fetus cannot, and it can result in, amongst other things, oh dear, I forgot the correct term, water on the brain. I'm sure someone will come up with the correct word right now. But people were becoming hydrocephalus. Thank you, hydrocephalus. So people were becoming very ill with toxoplasmosis. And another one that Cliff Jumper already mentioned was pneumocystis pneumonia. Pneumocystis pneumonia is really rare. It's caused by a fungus. So in the gay community, they were seeing these rare, and there were others as well, but seeing these very rare disorders. But it wasn't all the same disorder. Everybody didn't have Kaposi sarcoma. Everybody didn't have toxoplasmosis. Everybody didn't have pneumocystis pneumonia. So there was this suite, so to speak, of rare diseases appearing in the gay community. And this eventually led to the discovery of the HIV virus in humans and what they called acquired immunodeficiency syndrome, which is what basically the immune system took a dump and allowed these very rare things, whether it's a virus or a fungi or a bacteria or a protist, these rare organisms to take a hold in the body. And that's what eventually killed people, was HIV. But if HIV had entered the general population first, it would have been far harder to identify, because you'd have had a case of sarcosis over here. Kaposi's over here, a case of pneumocystis pneumonia over there, and it would have been very hard to draw the lines between them. So it was actually the fact that HIV entered the gay male community probably saved millions of lives. And the other two communities that it entered mostly were, of course, hemophiliacs, because they needed, blood material, and drug addicts. And there's a whole political spectrum to that I'm not going to get to right now. But I think let's go into having scientist Mel tell us what exactly is HIV.
Okie dokie. So HIV is not a fun thing to get. It's horrible in regards to not so much the infection itself, but what it causes in the long run in regards to completely debilitating your immune system. So you end up getting all kinds of different illnesses you wouldn't normally have because you have a strong immune system. So I thought I would talk a little bit about the genetics in regards to HIV, and also why there are some drug-resistant strains. Now, that has a lot to do with the mutations. HIV does have mutations. There's so much information available, and since we're not here for a super long period of time, I thought I would go ahead and show you a few things. First thing I'm going to show you is actually how HIV attaches to a cell, and it attaches to what we call a T cell. So I hope I can share screens. I think I can. Screen share. So I'm going to share this screen really quickly. Hopefully you guys can see it. I'm going to present to everyone, I guess. All right, so here.
If you press Control and press the plus key, that picture will get bigger.
Okay, it's here we go. So what we have here is an HIV virus here. This particular receptor is called a GP120, and it's attaching to this T cell. We have what's called a CD4. Now, the thing that's important about this is how viruses and cells communicate. So what happens at this point, and it doesn't show you in this particular picture, but this here is the genetic coding inside of the virus that will then enter into the cell. So let me see if I can find, I had a picture I wanted to use. Okay, let's use a better picture. Let's kind of get, that's not a good picture. I had a good, here's one down here. Now this has a lot of numbers on it. That sounds scary, but that's all right. I'm going to open image in new tab. I have like 9 billion tabs open. So there we go. So let's look at the breakdown of this. Okay, so here we have HIV, human immunodeficiency virus. This is kind of like the structure similar to the cartoon we saw earlier. Here's GP120. Now, GP, these letters right here, P, P, GP, this all has to do with the part of the coding of the virus that expresses this particular receptor. All right. Now, This would probably more than likely be HIV-2. There are 2 strains. HIV-1 primarily would use what we would call GP36 for detection. And here we have P24. All right, so I actually work in viral antigens, and I've recently purified GP36, and they use this for testing because those particular receptors are what antibodies kind of attach to. So for if you have HIV-1, for instance, wait, no, this would be HIV-1. HIV-2 is GP36. I got that mixed up. So this is HIV-1. It would test for P24. HIV-2 would test for GP36. So let's have a look at the structure. But I had the structure. That's a paper. That's not it. That's not it. Here it is. I'm going to open this in a new tab. And we're going to take a look a little bit about the difference between the 2 genetically. So here are some more scary letters. But this essentially, I won't get into what all of these mean. If you're curious, you can hop over to this website here, ibase.info/QA. If you just type in HIV genome explained, it gets into what all of those particular I would say acronyms. That's not the right word for it. The word escapes me. It gets into what a GAG is, what a POL is. And for instance, that's one of the reverse transcriptase. And I'm going to talk a little bit about what a reverse transcriptase is, because we kind of need to know that in order to understand the medications associated with antivirals. So if you notice, HIV-1 and 2, notice they have the same genetic makeup, right? They have all of the same stuff here. It's just they're arranged a little bit differently. Now, this is kind of important because that plays a role in how those particular receptors on the outside that we saw right here are expressed and how they attach to a T cell. All right, so that's kind of important. So I'm going to stop sharing now, although I really do like that GIF. That's a cool GIF. So I'm going to stop sharing now and kind of jump back on over here. Is my camera back? I don't know how to. Oh, there I am. So let me talk to you a little bit on how mutations work in regards to drug resistance. Okay. There is a database called NRTI. NRTI, I guess I could probably still share screens, but it's got a lot of letters on it. And this is run by Stanford University. They have an entire database that's just for drug-resistant HIV strains. And if you check it out, they keep up with all of the different strains of HIV that are resistant to particular drugs. And these mutations, a lot of them have to do, there's different ones. There's called TAM patterns. That's a thymidine analog mutation. And so they're selected by thymidine analogs AZT and D47. Now, AZT, you might have heard of AZT in the news. AZT is a type of drug that stops something called reverse transcriptase. One of the things that makes HIV inherently problematic is the fact that it can take RNA. RNA is what we call ribonucleic acid. It's just like a single strand of genetic code. It's not double-stranded like we have in our cells normally, inside of our nucleus, right? We have double-stranded DNA. Well, what makes HIV inherently problematic is that it can convert RNA into DNA and stick it right back into your nucleus. That's why it's called a retrovirus. It's able to take RNA, put it back into DNA form, and then it introduces it back into your nucleus. Well, guess what? Your nucleus, inside your nucleus, your biochemistry is so incredibly awesome that it's like, hey, look, here's DNA. Let me incorporate it into our genome, because how did it get there? Something wonky's happened. So it ends up incorporating that viral genetic code into your DNA. That's why once you are infected with HIV, it never really goes away because it's permanently incorporated into your own genetic code. And so anytime that particular gene is activated inside of your nucleus, it starts to make proteins associated with HIV, GP36, P24, and all of the different components that make up the virus. So this is one reason why once you have it, you kind of have it forever. Now, the thing that's kind of hopeful, AZT stops the reverse transcriptase from working. So it kind of binds to the RNA that HIV molecule pushes into that cell. It binds to it and prevents that reverse transcriptase from working. So I have really cool things called crystal structures. I love them. I used to work a lot with them when I was working in computational chemistry. So I'm going to share screens again, and then I'll show you kind of what that looks like. Let me pull up. Now I'm going to show you before AZT, what reverse transcriptase looks like, and after AZT, after it's like messed up. That's not it. That's a different one, but I'm going to downsize that one because I'll need that in a minute. Where did it go? It just went away. That's not very nice. Hold on. I downloaded it. All right. So anyway, let me see if I can find it. Where's the back button? I'm not very good at stuff sometimes. So
this is the reason why when I want to screen share for people, I normally do the screen sharing. So I basically say, okay, what do you want sharing in what order? So I can just chuck it on there and it's there. So you can do the presenting and don't have to worry about finding things.
Yeah. Well, okay, so that's after, and here's pre. I got pre again. All right, so I'm just gonna like pull this up on here like this. All right, so share right there. So this is a pretty cool database. All right, so here, I don't know if I can make that bigger. I can download it. This is what reverse transcriptase looks like before it's affected by AZT. So I think I can pull this up on a bigger one. You see it's all ribbony. All of these are different proteins, and these proteins attach to the RNA and work together to convert that RNA to DNA. Now this is what it looks like after it's been affected by AZT. Now the thing that makes this, can everybody see it? Or was that presenting?
Yep, we can see.
Okay, so this is before. So this is before, this is reverse transcriptase. So this is what attaches to RNA and converts it to DNA. Now, after it's been affected by AZT, notice the structure changes, doesn't it? Mm-hmm. This is very, very important because enzymes, if it ends in ASE, that means it's an enzyme. So polymerase, ASE is an enzyme. This is reverse transcriptase. The shape of enzymes are inherently important. So when it goes...
Can you just tell me what the differences in the color are? Are they just different strands or...
These, okay, so this is put there. It doesn't actually look like that in far as colors. But if I were a scientist and I wanted to research a particular strand, I could highlight on this. And this particular amino acid sequence, like this little orange, is going to be a little bit different from this blue. And so it's mostly put there to try to help. And if I still had my molecular orbital software, my MO software, we would be able to twist this around in different ways and kind of look at it fully in 3D.
Yeah,
so if you're...
Obviously, I know that DNA isn't color-coded. I just meant that, you know, obviously the colors are a symbolic of something, and they're just different,
well, this
different
isn't,
strains, did you say?
This isn't the DNA. This is actually the... enzyme itself.
All right. Okay.
Yeah. So this is the enzyme that attaches to RNA and converts it to DNA. So that's what that is. So if you want to take a look, this is pretty, the protein database is really cool with that. So I was going to show you really quickly. This is the HIV drug resistant database, and they go through and they do a really, really, really good job you can go to the crystal structures, and it lets you know all of the various mutations associated with this. We have TAM, we have TAM has to do with thymidine and changing that out. There's multinucleoside RT inhibitor resistance mutations. I know those are all like really scary sounding words, but if you want to dive down deep into this, this is an entire database of all of the various mutations associated with HIV and the drugs that they're essentially resistant to because of their genetic mutations. All right, so one last thing, and then I will hand it over to the next person. So the things that's pretty tricky and something that they found out recently, depending on which receptor that HIV attaches to, also is key in regards to the severity of the progression of the disease. So HIV normally attaches to, let me find it, it was a CDR5. Here we go. Yes, so it normally attaches to the CDR5 particular receptor that's on the outside of T cells. Now remember, receptors are there and they're how cells communicate with one another. Now what can happen is it's the CXCR4, that's the other receptor. And so certain strains of HIV, about 50%, can switch between the two. Now, if it switches over to that X4 receptor, there tends to be a faster progression of HIV infection than if it's in CD5, that CDR5. So, so they're trying to figure out right now, scientists are trying to figure out, and there's this particular mutation associated with that allows HIV to be able to switch between those receptors. So if they can figure out what that is, they can possibly shut it off from going to that more dangerous X4 to just keeping it to the regular CDR5. So that way, if they're able to kind of implement that and stop it from switching to that X4, that can at least slow down the progression of infection. There's so much information on the genetics and the genome in regards to HIV, but essentially mutations, the virus can mutate pretty rapidly. Like with AZT, that was the very first drug that they had, and it has to do with adenosine triphosphate. And so by removing a little bit of that energy molecule, it's able to kind of bind to that RNA, remove that energy molecule off of the reverse transcriptase, then it doesn't function so well and it changes shape. But HIV, because it can mutate quickly, developed a strain to where it could use ATP and keep it from getting picked up by the AZT and causes it to be non-functional. I don't know all the molecular mechanics behind that, and I don't know if that's something that you guys would follow so well, but it's still considered a good medication that they use, especially with pregnant women who are HIV positive. It slows down any kind of transmission to babies, so it's still successful in that regard. So that's all I have on that. I have lots and lots of sources. So if you want all my sources, hit me up on Twitter or Google Plus, and I will just send them all to you.
Well, and under the video, if you want, you can chuck in a whole bunch of your links so people can go and check them out.
Okay, great.
I've already added in those that were in the side chat. They're already in the description. I've just put them in there, okay?
Dreadnought asked the question asking, could you speak on the double-stranded versus single-stranded virus comparison, specifically the fidelity involved in some of them being made of the antisense strand, not the sense strand? I don't know.
On HIV doesn't have double-stranded DNA or anything like that. It's all single-stranded RNA.
Do you want me to talk about that?
Yeah, we're talking about the fidelity, comparative fidelity.
Comparative fidelity.
Yeah.
Yeah, I think he just means
between
viruses in
DNA.
general.
Sorry?
I think he just means viruses in general, doesn't he?
No, he's
Yeah, that's what I think the whole point was.
on you go, RSC.
Because
Yeah, yeah.
Go ahead.
okay, I just wanted to clarify Drag's question. He's asking about what makes HIV different, because HIV is a much more different virus in terms of the genetics and how it's transferred, how it replicates in a cell. It's specifically what's called a retrovirus, and it's an RNA retrovirus. So we have
Right.
viruses that are either DNA based or RNA based. DNA based viruses use the same cellular mechanics that your normal cells use. They inject their DNA, the DNA gets translated into RNA, transcribed into RNA, and then the RNA gets translated into proteins. RNA viruses have kind of co-opted that system. They have RNA in their virus particles that gets injected into the cell, and they use a special enzyme called reverse transcriptase that does the backwards transition from RNA to DNA. And that's what they use to propagate their proteins and build more viruses in the cells. Did you want to add anything to that, Mel, or Jade, or Fiona?
Can I ask a question related to that? Are RNA viruses quicker at mutating, or are they about the same?
I would dare say they probably are quicker.
They are
because
quicker.
of the fact RNA has a very short half-life in the cell cytosol of a cell. And since you have reverse transcriptase, that's reversing, it's converting this RNA into DNA. You do have a higher rate, or a higher possible rate of mutation. So you can have a different nucleotide kind of pulled right in there as it's going through, and turning this single-stranded RNA into a double-stranded DNA molecule. And then when that gets incorporated into your genetic code inside the nucleus, because your enzymes are going to pick that up and it's going to incorporate it into your genetic code, it's got to do that somehow. Then you have an increased chance of additional mutations within that process.
One thing I've heard about, oh, sorry.
Oh, no, I'm done.
Oh, no, I'm just going to say one thing I've heard about is some people with HIV, just at that level, they can't keep it under control because it mutates so much, and they have to keep switching regimens. And then they came up with saying, well, don't take any drugs for a while. It's complicated. I can't explain it, but then they start them back again, and it seemed like it helps them. But it's like they're in a race. Can the medicine stay ahead of the mutation rate?
Yeah.
And in other people, it doesn't mutate at all. They take the same medication for years.
One of the things that causes HIV to have such a high mutation rate is because the reverse transcriptase does not have any way of correcting errors. There's no exonucleolytic proofreading.
Yeah, none. That's true. There are none.
And so therefore, you find with retroviruses, you get a much higher mutation rate than you would normally get. I remember being in a talk that was being given when I was doing my PhD. And it was the third year talks, so it's the final year PhD talks. And there was a guy there. He was an external PhD student who was doing his work in one of the local hospitals, but also through the university. And he had been looking at HIV. And it was thought at the time that what happened was that the virus was converted to DNA, spliced into the genome, and then it remained doing nothing in a sort of dormant manner until something reactivated it. And he studied some HIV patients. And he was looking at the number of free viral particles that he was finding in the blood. And I think he also looked at mutations, but I can't remember offhand. But from his numbers, he discovered that even in those healthy people, there was an absolute viral storm going on. And the room just went silent when he presented this data. This was a PhD student. He was saying, no, this isn't latency. It's not dormant. It's there. And it is replicating at a great rate. And it's just that it eventually exhausts the immune system. Because remember, it's immune cells that it's replicating in. It causes the immunodeficiency eventually because it's immune cells that it's ending up killing. And you could have heard a pin drop when he presented that data. That was the first time any of us had heard that concept, that this virus was there and replicating like bilio, and you didn't know it. You had no symptoms. And that it wasn't a dormant virus. Sorry, I just thought I would throw that in there because, yet again, it's one of those, I remember the guy standing there with the remote control for the projector, and he put up the slide with the data on it, and everybody went, oh. That was a surprise.
Fiona, I'm sure.
What's a dormant viruses like? I mean, do they just sit there and do nothing particular?
It's like cold sores. You never get rid of a cold sore. Chicken pox. If you've had chicken pox, chances are you'll get shingles later because you still have the virus there. Right. So you're talking about dormant then. They're not doing anything and they can reactivate. And they thought the HIV was doing that in the form of double-stranded DNA within the genome. It was just sitting there and doing nothing until something switched it on.
Right.
And this is why it took so long to show up. But his data seemed to show that no, there was ongoing viral replication right from the word go.
Now, do you mind if I pick up from there, Fiona?
I just want to ask.
Does anyone have questions, Cliff?
Yeah, I got a very, very quick question. When you said it spreads rapidly, how long? Hours, days, weeks, months, years before it's something that's really picked up?
Well, it's when you get the symptoms.
Yeah, the time from infection, introduction to the body, to the time you start showing symptoms will vary from person to person. It really isn't. I mean, if you're a very sick individual, you're much likely going to show more rapid progression. But just because you're very fit doesn't necessarily mean that you have some protective effect. It's not anything that could be forecasted, for lack of a better word.
Right.
Yeah. But it's at this point I want to ask Heavy Hauler to give us some information about HIV from an eyewitness perspective. We've taught the science here. And I want you to all remember that when this first began, people with this disease, they were shunned. They were treated very, very badly. We all know about that. But while they were being treated like that, in such an awful way, they were also having to deal with other aspects of the disease, the actual manifestations of it. And Heavy will be able to maybe explain some of that to everyone and why we really should be, even now, still ashamed of the way that HIV and those infected with HIV were treated in the past. Heavy, on you go.
Well, for me and my partner, it wasn't that bad. But, we heard stories about other people, how they were treated when it came out that they were, HIV positive or they had AIDS. My partner and I, he was diagnosed in about 94. Yeah, 94. And I kept holding off on getting, checked out because we had been together for a good many years before we found out, because we had early testing, both of us, and the test kept coming back negative. But it was back in the days when the tests were not that good, and that's kind of how the blood supply got infected. And hemophiliacs were catching it because the tests were missing the virus that was there. But anyway, he got real bad sick and was in a induced coma for about a month to clear his lungs out. Now, he was a big dude. I mean, big, burly man, very strong. But it knocked his feet out from under him. But I kept holding off on me because I was afraid of what might happen when my insurance found out, or people at my job that handled that kind of stuff. So he lived for 3 years after that, but it was pretty rough because he got weaker and weaker and just wasted away. He wouldn't tell his family. They didn't find out until after he was gone, and it really shocked them. It really did. So he was very fortunate in that we got in touch with, you could say, the right people. There's a group here, we're down in Charlotte, and they help AIDS patients. And they find resources to help, And then the hospice came in. And hospice, those are some amazing people, very knowledgeable, very caring, very loving. And there was even a church group that would come over and they would bring dinner and stuff like that and come and see him. And sometimes they would take him out out shopping or something when I was at work. But he eventually passed away. Now, and then about that, just before he did, I finally broke down and went and got tested. And I had lost a lot of weight. I went from about 200 and something down to about 150. Of course, that could be from stress as well, working, taking care of him. And as soon as I started taking my medication, it turned around and my viral load dropped. My CD4 count came up. I have been HIV positive for just over 20 years, and I'm doing fine. The medication agrees with me. So far, I have not been. Oh, bless you.
Well, that just scared the life out of everyone.
I'm sorry. Apologies to anybody through headphones. Oh,
well, I was putting everybody to sleep, so I guess you're free to wake everybody up. But
I'm sorry. And the problem is,
but I
wanted to point away from the microphone, but couldn't to mute myself at the same time.
But like I said, I've been very fortunate. No side effects from the medication or anything. CD4 count still like it's undetectable. I mean, my CD4 count's good. Viral load undetectable. My HIV specialist I went to see, he said after about 2 years, about 99, he said, "You don't need me anymore. Go see a regular doctor." I hadn't seen him since. But they still run the test and send them to him, so he can check them out. So HIV, I mean, the virus caught my partner by surprise. Well, caused both of us by surprise, but it floored him. I mean, it was like it just knocked him for a loop, and he never recovered. But it was different for me. It didn't do that. It was more, I guess, just slow. It was there, but fortunately, I'm still here. And I never could understand that because he was so much stronger than me physically and mentally in a lot of ways. But it hit him like that, and then me, it didn't hit like that. And well, here I am.
I have a question. Did you? I didn't hear. Did you have the same? I would imagine you had the same strain as your partner, but it's possible that you could have had different strains.
I don't know. Well, see, we've been together for, oh gosh, about 8-9 years maybe before this happened. Before he came down with it, it just hit him. His lungs started filling up very fast, and he couldn't breathe, and we had to go to the hospital.
Question.
Before that, there was no symptoms. There was nothing.
Did they ever analyze? Did they ever tell you which strain you had or which strain he had?
Not that
One or
not that I
two?
recall. Because see, I
I
kept
think that was before that
putting it
test was
off.
available.
He passed away around the time that I finally broke down and went to get tested.
Oh, I see.
So there was no comparisons. I was, like I said, I was afraid what might happen for my insurance and stuff like that. But I'm very fortunate. There are groups out there that help people that may have trouble affording the medication. Because when I first started taking it, it was like over $3,000 a month for my medication, and I didn't have that money. Excuse my language, but there were groups and organizations out there that help, and even now I still get help from the drug manufacturer, and also from another group. Oh gosh, I can't believe I forgot their name. Copay Relief. They help.
I have another question.
Yes.
How quick between the time that he was diagnosed with the infection do you think it was until the actual onset of the
Well, so he wasn't diagnosed.
secondary infections?
He wasn't
wasn't?
diagnosed. No. See, we had tests. Like I said, we had tests in the early days when we first started getting together and we started living together, and the test always came back negative. But see, he worked for the Red Cross, so he was up on a lot of this information, and he said that like some of these tests, later, he said, when after he found out that he had it, he said like, the early tests were not that good. And so we stopped getting tests because we were monogamous. We were in a monogamous relationship. We didn't, play around with stuff. So we figured, yeah, we're safe, and it was like out of the blue. Like I said, his lungs started filling up. He couldn't breathe, and they had to put him in an induced coma for a month. Like the next day. I mean, it hit him that hard and that fast. And so after that, it was downhill. So, he would get maybe some secondary infections or something. He was losing weight. Well, he lost a lot of weight that first month when he was in that coma, but he never recovered. He couldn't work or anything. They put him on, SSI, I think they call it, to help out as well. So there's no way he could work or anything. So there was no diagnosis. It was like, bam! It was like getting hit by a Mack truck.
So all the secondary infections started hitting at once.
Yeah, it was like the lungs filling up with water. And that's
so that was pneumonia. Then
we thought it was. I mean, it was. I guess whatever they call that, where the lungs fill up because of the virus is causing it. But it was like he was full blown. We didn't know it, and it just bam, like a Mack truck, and that was it.
That'd be pneumocystis carinii.
Yeah.
I got a question, not for heavy at all, but more the sort of general, how it's dealt with. Do they keep samples from all HIV/AIDS patients to keep a track on the disease with the goal of fighting it?
It probably goes into some kind of database, I guess.
It's a reportable disease, and results will get reported. Samples probably are no longer used, but back in the earlier days, yeah, I'm sure the CDC and the NIH requested samples for everyone who tested positive.
Yeah. Now, like I said, we heard about people that turned, some of our friends that were positive or did have AIDS, and how they were treated by other people, and it was horrible. But we were fortunate in a lot of ways, and I'm still very fortunate. I mean, my doctor said my only problem is high cholesterol, and I need to lose weight. I was up to almost 250 pounds.
Wow.
And I came down about 15 pounds. And he said, you need to lose weight for health reasons. But if someone comes up positive, it's not a death sentence. It's not the nightmare that it used to be. The medications, most, and in my case especially, the medications just stopped it cold. I mean, it's still there. It's still doing whatever it does, my body. But there are no side effects, no secondary infections. There's nothing to be around me and to see me doing stuff. You would not know. that I was HIV positive and have been HIV positive for over 20 years.
Wow.
So, you know, I mean, that's kind of a word of encouragement for people as far as their physical health, but as far as how the family might react or their coworkers and stuff, well, I don't know. There are people out there that will help you. You know, I mean, I'm just kind of talking to the audience in general. If you find out you are positive, there is help out there. There is counseling. There are groups out there that will help you with your medication and treatments. So, you know, so it's not like the end of the world. I mean, it certainly wasn't for me. I mean, for me, I lost my partner. But yeah, it's a good thing it was about 20 years ago, because it would have been hard to talk about. But
I got a question.
That's pretty much about all I have to say, really. The only other thing I was going to add was, you know, like I mentioned, there are some people that the virus runs rampant. They can't keep up with it because it mutates so much. I've never had that problem. I mean, that may change, you know, next year or something, but no, mostly I just want to give a word of encouragement for people that, you know, if they do find out they're positive, there is help out there.
Got a quick question on the end of what you were saying. Does I know that the American insurance system is, you know, some people don't qualify and, you know, whatever, they don't get covered. How is HIV when it comes to health insurance companies?
Well, the insurance company I'm with, when they, you know, of course they found out, because, you know, I needed medication and stuff. I had like a deductible I had to meet, and I met it, you know, and I got help with paying for my medication. Once I hit my deductible, even though I was HIV positive, my medications were over $3,000 a month that I'm paying for. After I hit my deductible, insurance takes over. So I would hit my deductible in about 3 months. So for the rest of the year, my insurance took up the cost of the medication, even though it was like over $3,000 a month. So,
that's one of the things that really bothers me about the American healthcare system. Yeah. Here in the UK, we are fortunate that we have a national health service, and, you know, effectively medication is free, so to speak, to the point of use. You know, but it just bothers me that, you know, poor American who happens to contract HIV can't get medication.
Now, if they don't have health insurance, I don't know. You know, there's bound to be something out there,
right?
I mean,
I can talk about that. How uninsured people, I get them medication. But first, I want to say, Heavy Hauler, thank you so much for sharing that story.
Yeah,
it's really meaningful to this conversation.
Yeah, I agree. If I want to add on what he says there, it's true, because, you know, what we were talking about until then was very clinical. You know, it wasn't about people as such, you know, and the human touch is something that gets missed in these discussions, I think.
Yeah, that's why, I mean, when Heavy asked for the link, I was more than happy to let him in here, because we do discuss the science quite a lot. But this is one of the ones where, with there still being a bit of a stigma attached, people need to understand what this means for people that have the infection. That if you stigmatize them, if for some reason they want to avoid having to get tested. I know when it all began over here, people wouldn't get tested because just by being tested, your life insurance company would then assume you were in a high-risk group, and your life insurance would be cancelled. So you could not be tested. That's not medical insurance, that's life insurance. And I also know of one case where a lab worker was exposed, and it was samples coming from the drug addict antenatal clinic and the lab worker was exposed, and they would not release the information whether the patient was HIV positive or not, because it would stigmatise the patient. And the only thing they would offer the lab worker was, we'll take blood from you now, and if you get ill in the future, we'll take another sample of blood from you. What use is that? And it was because of all the stigma that surrounded this disease, we ended up with people being exposed without knowing they were being exposed. We had some doctors wouldn't label specimens from patients as a high risk or an HIV risk because it stigmatized the patient. But that then left the lab workers being exposed without knowing it.
Yeah.
Whereas if a patient had something like tuberculosis, all of their samples would be labelled as high risk because tuberculosis didn't carry the stigma. So that would be put on it. If they suspected Legionella, they would tell you that.
Mm-hmm.
But when it came to HIV, some doctors would refuse to say, even that it was a category, it was Cat B at the time, that was a category B sample. They wouldn't even tell you that, let alone that it was an HIV risk. And in those days, when your samples came into the labs, you didn't wear gloves when you were taking all the samples out the bags. And if they were leaking, you were exposed. But if it had the sticker on it, you did wear gloves and you sorted all the sample out in a laminar flow hood. So you needed the heads up. That didn't happen. And again, it was because of the stigma that was being attached. And doctors did not want to stigmatise their patients by labelling those samples. And I think doctors were really stuck between a rock and a hard place, because they're not responsible for the stigma that was attached to it. And they have to have the best, their patient's best interest at heart. But then those lab workers at the other end, what about them? Who's looking after them? And it became a real problem in trying to work out a policy whereby the lab workers would be protected and at the same time patient confidentiality wouldn't be breached. And yet, every time you send a sample to a lab, on the little form, you put a brief medical history so that the lab knows what they're dealing with, but not with HIV. And it all came down to that stigma that was attached to it. And hearing, Heavy saying that he was avoiding getting tested because of this sort of thing. And insurance problem and all the rest of it and his work. And with a disease like HIV, the quicker you are tested and on treatment, the better. And yet society made it that you wouldn't want to get tested. And it ended up with so many more people ending up sicker than they needed to be. I find it very, very sad. Heavy, I want to say thank you very much for sharing your story with us. And I was going to now ask RSC to hit on the medical side of the disease.
Can I just say something very quick before he does? Someone asked in the side chat, because I mentioned about the medications and things like that, in the US healthcare system. They asked, medication in the UK is covered by the government? Yes, it is. People in work pay a small amount, so £8. It's currently £8.80 per medication a month, or
not in Scotland,
isn't it? Not okay. Oh yeah, because they removed it,
in Scotland, all prescription medications are free.
Yeah,
utterly free.
I wanted to add briefly,
but it's £8 in the rest of the UK. It's £8.80 per cost per medication, so for a month or whatever. No matter the cost of the medication. So if it's £1,000 a month, you pay £8.80. That kind of thing. Poor people out of work or on Social Security, they pay nothing. So.
Right.
Go ahead, RSC.
I know Jade wanted to chime in with something, but if I could just pick up with the American side, people who don't have health insurance, they have to get their medications through pharmaceutical group, some kind of charitable organization. Luckily, there are so many organizations out there that are sponsoring that kind of work in an effort to eradicate HIV. It's fairly easy to find people who don't have insurance, those medications, even if they can't afford to pay it. Jade?
Yeah, okay. Can you hear me now?
Yeah.
Okay. So I just wanted to briefly add to what Fiona was saying. And one of the problems we had here was with emergency workers. Because emergency people would respond to various things, and they would have no idea if the person had HIV or not. And if they were, and I'm sure RSC will go into how one acquires the HIV virus, but if there was a chance that they might be exposed, they couldn't find out later because the hospital would not tell them whether or not the patient was HIV positive or not. The other thing I wanted to mention is here in the San Francisco Greater Bay Area, during that time, there were confidential clinics that you could go to get HIV tested. Because of this stigma, because people did not want to go to their primary care physician or use their insurance for fear of what Fiona said, that they would lose their insurance, including their life insurance. They might lose their job if they were found out to be HIV positive. So we had clinics here that were confidential where people could go and get tested. And the only person who would know that they were HIV positive was the clinic and themselves.
Thank you for that, Jade. So I'm just going to pick up real quickly with a little bit more about the basic biology and microbiology about HIV. We talked about that there are two serotypes, HIV1 and HIV2. And again, I just want to emphasize that these are retroviruses. They're members of the Retroviridae family, the genus Lentivirus. And we talked about how retroviruses, they use RNA and they go backwards to DNA, which makes them different from all other viruses. But some other characteristics about HIV that are important is that it's an enveloped virus. Much like influenza, when we talked about influenza, that's also an enveloped virus, which means the virus is coated with the outer layer of your cell. It's called the cell membrane. So the virus basically steals a part of your cell's membrane as a cover. And that's part of the reason why it's very hard for the immune system to detect the virus, because it's basically wearing the skin of your cells, for lack of a better term. HIV is also a diploid, which means it carries two copies of its genome. It is a single-stranded positive RNA virus. And that just tells you which virus or which RNA strain of the virus is the primary one for encoding genes and how those genes are read. So those are just some basic biology aspects about HIV that we didn't really hit on. But the reason that those things play a big role is it, like Mel was saying, that CD4 receptor is the receptor that certain cells called T cells use to basically feel and check your cells and check all foreign particles to recognize whether they're foreign or self. Since HIV has that coating on the outside, its envelope, it displays or mimics the signals that say not foreign, that I'm part of you, recognize me and don't attack me. So because the T cells read the virus outer shell like that, they don't get targeted. It doesn't get sent for destruction. It doesn't get attacked by the immune system, for lack of a better word. It's that CD4 receptor I've kind of described as like a Velcro type connection, where the virus can come up, attach to that receptor like a Velcro, and easily peel off. That CD4 receptor is kind of like the way the T cell feels around to distinguish things that are foreign versus self. It's not a very tight binding receptor. And then scientist Mel talked about some of the other receptors that are involved. Well, if the HIV is going to transmit its RNA into the cell, it can't rely on this Velcro, this weak connection receptor. So there are other receptors that have to be involved. She talked about CDCR and CCXR. Those are other ones. Those are a little stronger connections. If the virus can connect those receptors also, it can pass in and infect that cell. And you only find these receptors on these CD4 T cells. So that's why they're the main targets of HIV. HIV can easily attach to them with that Velcro-y type receptor that's everywhere in the body. And then it can use other receptors to give it a firmer attachment and then actually infect the cell. The sad thing is that the CD4 T cells, they're the communication hub of the whole immune system. They're the ones that will talk to the immune cells that are everywhere, that are part of your different immune systems that we've talked about in the past. You have your always present immune system, and then you have the activated immune system that gets kicked on in response to infection. And I'm trying to use common language so that everyone can kind of follow along. So the CD4 cells are the ones that do the communicating between those two branches of the immune system. When you knock them out, everyone's basically flying blind. And that's why HIV is such a bad disease to acquire, because once the number of CD4 cells falls below a certain level, you become susceptible to infections that you would never in your right mind think you would get, because they don't happen. Things like toxoplasma, those don't happen in healthy people. Things like CMV virus, you don't see that in people unless they're immunosuppressed. Pneumocystis jirovecii, that fungus we talked about, people who are healthy never get that infection, never need to be treated for it. And actually that's the one that the medication where that one guy who went to jail recently, he jumped up the Daraprim price by like 200 or 300% or 1,000% or something.
Pharma bro.
Pharma bro guy. Pneumocystis, that was the medication used to treat that condition. And Heavy Hauler mentioned thrush also. That's one of the things you'll see in HIV positive patients when they get the AIDS label. The AIDS label happens when your CD4 count falls below a certain number, when you're susceptible to those infections that healthy individuals will never be susceptible to. So let's talk about how people acquire HIV. There's basically 4 ways that you could acquire HIV. One is through blood contact. So we talked about that the cells that are mainly affected are the lymphocytes. They're in the blood. If they get into your blood, that's how the virus can transmit and infect cells in your body. So blood can be just direct contact with blood, but also through sexual contact. The second way that people get it is, the second most common, is mother to baby. HIV does cross through the placental barrier, and babies and fetuses, unfortunately, are very susceptible to that. Even with a proper medication regimen of like AZT and ZDV, transmission rates can get to 3 to 5%. So even if you take care of the mother and do everything that you can for the baby, deliver by C-section, make sure the mother is taking the meds and having her HIV count low, there's still that possibility that the baby will be infected when they're born. Also, things like needle stick, tattoos, needle sharing, drug use. Those are the other mechanisms that are pretty frequent for HIV transmission. It's funny we're talking about this today because we actually had a lecture about sports and the topic of HIV came up. How do you clear patients to play in contact sports? Well, I think there's only ever been one documented case of someone playing a sport transmitting HIV to someone they were playing against. It was in a soccer match in the UK. So of all the millions of kids, adults, professionals who have ever played sports, HIV has only been transmitted one time. So if you're playing sports, you're not, like, even if it's contact, wrestling, karate, things like that, there's very low likelihood that you can get HIV through that. Breast milk, though, that is infectious. Saliva, not very infectious. It's very rare to get from kissing someone or having spit on you. That would be much lower than other forms, but sexual contact and blood contact are the two highest, and then mother to baby is the second highest. Were there any questions about how HIV is transmitted?
No.
There was one thing I want to bring up. If I recall correctly, if the virus doesn't have a medium to sustain itself, like blood, if somehow it gets on something, it doesn't survive good outside of the body at all. If I recall.
A classic example I would say is a toilet seat.
Yes.
From a toilet seat,
Oh gosh.
it dies when it gets exposed to the outside environment, just like Heavy said. If it's not in a medium, it doesn't survive. It becomes non-infectious anymore. I did see one question from Benna. He was asking if HIV causes inflammation. No, HIV doesn't cause inflammation. HIV basically degrades the immune system's ability to communicate between the two networks. You have your two immune sides, one that's always present, always active, and the other side that responds with antibodies. When they can't communicate, there's no coordination. The ability to fight infections, it just becomes very ineffective, and you can't fight off the very basic things. So it's not the HIV necessarily that causes the inflammation, but the HIV allows for simpler infections to come in. So you can get a pneumonia. You can get a urinary tract infection, and your body doesn't have the mechanisms to fight that back, but it normally wouldn't be a problem. And because your body can't fight it back, a lot of people will pick up a disease and carry it for a long time before they develop any symptoms. The symptoms you get when you become sick are mainly from your immune system fighting the infection. And if your immune system's so weak it can't fight, you may not develop any symptoms. You may have a pneumonia. You may have an abscess somewhere in your body that's not being defended against. So you don't develop any symptoms and you don't notice anything. And sometimes it can hit people really hard, like Heavy said. And not to mention that immune system, when you do give people the HIV medication, a lot of people's immune system bounces right back. That can also not be a good thing, because if it bounces back too strong, you can get problems and symptoms and side effects from that. So, I mean, I'm very happy to hear that Heavy's doing so well. But I would say most people do very well on the medicines now. The problem of drug-resistant HIV is always going to be present, just like we have problems with drug-resistant bacteria. But just last week, they introduced a new FDA-approved medication for drug-resistant HIV. It's very expensive. I think it's like $300,000 a year, but if you need it, they're going to make sure you get it.
That's important.
One other thing, which I think is interesting about this, is that one of the things they'd noticed before they identified the virus was that there was one condition that seemed to be common amongst a lot of the individuals, and it was Kaposi sarcoma, which is a very particular type of malignancy. And it seems that normally Kaposi sarcoma is caused by a specific herpes virus, human herpes virus 8. And with most people, it doesn't cause any problems. But there are individuals that they will end up developing a Kaposi sarcoma. In patients who have HIV, Kaposi sarcoma can end up all over the place. And it's because, again, with the immune system being compromised by the HIV infection, the Kaposi sarcoma is able to take hold. And there's very little can be done about that. Many of the HIV patients have ended up with Kaposi sarcoma spotted all over their bodies. It shows you just how disseminated this human herpes virus 8 is, and how much your immune system is doing on a daily basis to keep it down. So that was, I think it was before they had a decent test for HIV, they were, the diagnostic criteria were basically pneumocystis and Kaposi sarcoma. So you were already in full-blown AIDS by that point. Which is rather sad.
Am I presenting
Yes,
a picture?
You are.
Just to let everyone know, this is an electron microscopy image of the HIV particle.
That is beautiful.
And obviously this is very magnified. So if I can give you a little bit of the scope or the width, I think this picture was pretty good.
That is a stunning picture. Oh, and there's a little, was it budding off from the actual cell itself?
This is the cell, the one cell that's being infected. And over here we have the HIV particles. You see how tiny they are in comparison. And even in comparison to other viruses like HTLV, which is a human T lymphocytic virus. I'm sorry, lymphotrophic virus. It's still a little bit smaller than those viruses. So we're talking on the microest of scales.
Yeah.
Which is why it took so long for them to figure out what it was.
Yeah.
Did you guys want to see pictures of Kaposi, or do you want to not gore the audience?
We'll leave those pictures out. They can look up the pictures of Kaposi sarcoma if they want.
All right.
They're not a lot of fun. What was I was going to say something. Yeah. I remember when I think, was it the Paris group that published the first electron micrograph of the virus? I think it was it budding off from a cell?
I think so.
That very first electron micrograph that they got. I remember that one.
Because, yeah, like I said, the virus has to encode itself with a part of your cell's membrane. So it buds off when it's fully formed and is ready to leave the cell.
Yeah. Now, we haven't really touched on a lot of the sort of the myths that surround us. There are obviously those myths that, oh, it was created in a lab. Well, bring us the evidence, because so far all of the evidence is that it is so closely related to the simian immunodeficiency virus that is where it came from, and came from bushmeat, as Cliff Jumper described earlier.
And they can follow the DNA track.
Yes.
The DNA history.
Yeah.
And they can follow it all over the world to the different areas, because, as I sort of mentioned in my little thing, there are different variations of it.
Mm-hmm.
So some parts are in East Africa and some parts are in West Africa and so on. So different versions. And, you know, they can follow the history, the genetic history through time. They even have, they found samples from like the 1950s and 60s. One of the examples I remember, let me just pull up my record thing, because something I found in one of the other versions. Earliest evidence of HIV in East Africa comes from Nairobi, 1980-81, where 1% of patients with STDs and 5% of sex workers were HIV-1 infected. Just 3 years later, 82% of Nairobi sex workers were HIV-1 infected. Most interesting is that of a Norwegian family, father, mother, and a 9-year-old daughter, who all tragically died in 1976 from AIDS caused by HIV-1 group O, and whose samples were found to be HIV positive when tested 12 years later.
Wasn't there a sailor and a young boy from the 1950s?
That's on the, yeah. The child was born in 1967, which implies that the mother was already HIV infected by then. The father had been a sailor, visiting a number of ports in Africa in the early 60s, where he did develop STDs, presumably after contact with prostitutes. He probably acquired HIV-1 in Nigeria or Cameroon when his boat was stopped for a few days in 1961.
Yeah.
Well, I'm going to hop off here. Thanks for having me on here. I have to go.
Thank you for coming, Mel. Thank you very much. Your information is much appreciated.
Hey, y'all have a great night.
Yeah, you take care.
Yes, thank you. Goodbye. Thank you very much.
Thank you, Mel.
Bye.
Bye.
There's another one here. A Danish surgeon died of AIDS in 1977 after working at the Abumon Bazi Rural Hospital in Zaire in 1972 and 75, and in Kinshasa in 1975. Following an early stint in archival samples, the same country around 1964, an 8-year-old Zaire child infected peritoneally. Is that the word?
Peritoneum?
Yeah. In 1974. Died in Sweden in 1982, and AIDS was serologically proven later on.
So, I mean, it's been around for a lot longer.
Yeah, I got one more on this, which is really quite sad. A very unfortunate Canadian pilot involved in a plane crash in 1976 in northern Zaire, where he had surgery and received a blood transfusion, died of AIDS in 1980. His serum was later found to be HIV-1 positive. Transfusion-acquired HIV infection progresses rapidly to AIDS because of a huge number of viruses present in the blood bag. So wow.
Yeah. So, I mean, it's been around for a lot longer than we were ever aware of it. And all this nonsense about it was developed in a lab. But if you look at the dates for where the people have actually got ill and died, and compare that to when DNA, the structure of DNA was discovered, then you'll see why this idea that it was manufactured in a lab, it's just so much marsh gas.
You know, I remember I had a conversation with a street preacher in Manchester one time, not someone that's on the internet, by the way. And he was a Episcopalian, I think it was. So it was fairly rare, I suppose. Anyway, so what happened was he was saying, "Oh, it was the gays," and I went through the spiel of how it started from the bushmeat trade, and he listened patiently, and in the end, he just said, "No, it was the gays."
Yeah.
Everything I said just didn't care. No, it was the gays.
Yeah, I wanted to bring up something about the transmission in Africa.
Go for it.
Yeah.
What did Freddie do now? Jade, are you still with us?
Have we lost Jade?
I'm afraid so. But Fiona, your point's spot on. There's no way for us to have created a virus before we were even able to discover what the virus was.
Yeah.
It's ludicrous, and people with backwards thinking like that need specialist help. So
yeah, they do. I've put a message in for Jade that we can't hear a word she's saying. Just silence from Jade. I don't know what's happened. One other thing I want to touch on is a myth, a very dangerous myth that is still out there. And sadly, the Roman Catholic Church played a part in it. When they claimed that the HIV virus could pass through condoms.
Yeah.
Now, they weren't interested in stopping people catching HIV. They were interested in stopping people using condoms. And they spread this idea in Africa that a condom would not protect you. It wasn't true.
I actually have another... Sorry.
The data that they were basing it on was so much marsh gas. But anyway, I wanted to give Jade a chance to do her bit, because we've been going now for quite some time. We'll be wrapping it up soon.
Can I add one more onto what you were saying there, Fiona?
Uh-huh.
There was another one. There's a weird local myth, wives' tale almost, that if you have sex with virgins,
Yeah,
that will clean you.
yes,
that will get rid of the disease,
Yes.
Well,
which has allowed a lot of.
Jade, you're back.
hello again. So where was I? Because that was
We never
very
heard
sudden.
anything. You didn't even get
Oh, okay.
started. Off
I
you
just
go.
want to say,
So
Just get Jade going while she's got connection.
yeah. So anyway, what I was going to say is that I wanted to mention how, and I'm watching my connection here. Behave. I'm surprised with this storm I haven't lost power, but seems to be everything okay on that end. How although here in the United States the HIV became kind of known as a gay disease because it entered the homosexual male population first. In many other countries, including Africa, it was primarily a heterosexual disease. And one of the ways it was transmitted, it spread in Africa, was along the truck routes. Because the men who would drive the trucks, delivering various things, various places, would often have a friend, a female friend, or maybe a male friend in different towns, different cities along the way. And those people might have other friends who drove trucks. So the HIV spread along the truck routes in Africa, it spread all over the truck routes. And sadly, there are a lot of HIV orphans, even today in Africa, whose, both parents have died of HIV. So I don't know if we want any questions about what's gone on so far, or should I go into my little ending thing about the politics and religion and stuff?
Let's check and see if there are any questions.
Yeah.
Live chat, any questions? Anything we can answer? I think we've hit everything they've asked so far. So I think, Jade, you should go ahead.
Okay. What I'm going to do is talk very briefly, and believe it or not, it is going to be briefly for me because I wrote it down. And I know that some people might want to add to it later. What I want to do is I want to talk briefly about politics, religion, ignorance, fear, prejudice, hatred, social change, pseudoscience, and AIDS or HIV denialism. That's a lot of stuff. And those subjects are worthy of hangouts of their own. So there's no need to go deeply into them, just to do a quick overview. So because this was looked at as a gay disease, politicians dragged their feet on addressing HIV. Religious leaders were seen in the media claiming that this pox, as they sometimes called it, was the deserved result of an immoral lifestyle. What AIDS was and how it was transmitted seemed very mysterious, and this led to a lot of misinformation that generated a great deal of fear. There were caregivers who were afraid to change the diapers of babies who were HIV positive for fear of contracting AIDS. The deeply ingrained prejudice against homosexuals that had already existed fueled this fear and exploded to outright hatred that was expressed not only in the media, but in an increase in gay bashing incidents. Now, ironically, those attacking homosexuals didn't realize that they might be exposing themselves to the virus. And there is at least one case that I know of where a gay basher, contracted HIV that way. However, as the plague expanded, voices arose. And people like Ryan White, who was a boy who contracted HIV through a blood transfusion and developed AIDS. And there was Paul Michael Glaser, whose wife also contracted HIV. So you had these examples, just regular people and celebrities. And a lot of celebrities chimed in to try to attack the fear and the hatred. And this includes Elizabeth Taylor, who worked valiantly for many years to fight the stigma against HIV. A former daughter-in-law of hers also got AIDS. But it really hit home for a lot of people in this country when they started to discover that they had relatives who had HIV. Relatives who'd been in the closet, who hadn't told them anything. But when they became sick, sometimes the families had to come in and take care of them. I had a cousin who died of AIDS, but his mother, up until the end, insisted that it was a blood infection, even though everybody in our family knew the truth, because she was afraid that people would judge her son for contracting HIV. And even as some religious people condemned those who had AIDS, others, such as the Metropolitan Community Church in San Francisco, reached out for the sufferers, and there were lay Catholic religious, and these are people who they take religious vows, but they don't belong to a religious order. So they're considered lay Catholics. And there was a group of lay Catholic religious here in the San Francisco area who adopted some of these HIV positive children who'd been abandoned because they were HIV positive. And that was a very sad consequence of this disease, that people were afraid of babies. And like many diseases, HIV and AIDS, and as I said, AIDS is a result of HIV, and the lack of misinformation and the misinformation led to sometimes wild speculation that opened the door for quacks of all kinds and all kinds of crazy alleged cures. And of course, that didn't end well.
Jade,
And then finally,
side.
What?
Have a look at the side, the last thing I posted.
Okay. I was just going to say, finally, there's AIDS denialism. The idea that this is somehow a man-made disease developed in some laboratory in order to control the population or certain segments of the population. AIDS denialists refuse to understand that not only does HIV have a long and traceable history, but the knowledge and technology did not exist at the time to even create such a virus. Point out that yes, in the UK, Princess Diana was a key celebrity for raising awareness that touching HIV/AIDS people weren't going to give them the disease. I remember that, she would hug people, and there were other people who would do the same thing. They would hug people who were HIV positive, and those pictures would spread around the country. Another thing this did, sadly in a sense, was it helped change for a lot of people their ideas towards euthanasia. This disease was so terrible, or suicide, I should say, more than euthanasia, that some people just decided that it was time for them to go. And they would have, at least in the San Francisco, area here, I know, and other places, when people decided they'd had enough, were wearing down, and they were tired of the repeated trips to the hospital, they were tired of the pain, they were tired of being unable to do anything, this was going to end in death, they decided to choose their time of death. And sometimes they would have a party, and all their friends would gather to say goodbye. And then they would take a cocktail of drugs and leave. And I know for myself and for a lot of other people, this changed the attitude toward people deciding to take their own lives. Or in some cases, again, with euthanasia, there were people who asked their partners, if I get to this point, will you help me? And this happened. When you see people suffer for so long and so horribly, some people had it better than others, and some people, it was terrible. It tends to change your mind about things. So did anybody have any questions about that? Because I have one little interesting thing at the end here, but if anyone wants to add to what I've said here, please do so.
I just want to say that these suicide parties, that's quite chilling to hear.
Yes, but they were glorious. I never went to one, but I had friends who went to one, because people got to say goodbye to the person while they were there. And the person got to know that they were loved. Because I've seen actual video of some of these parties where people were hugging the person who was about to leave. This person was going to die anyway. And yes, it is chilling. But it was also beautiful, because they had this one last chance to be with their friends. And their friends had this one last chance to say goodbye. And so often, people don't get a chance to say goodbye.
And I suppose they also get to die on their own terms as well.
Yeah, they got to die on their own terms. And, if a person dies in a hospital, well, hospitals tend to frown on parties that involve, marijuana and booze and other things like that. So they could have whatever kind of party they wanted, So yes, it was, it's difficult, but it's also very beautiful to be able to do things on your own terms, to have your friends around you. Although I want to be clear, most of the time when the very end came, the friends left, except perhaps for a partner or a close family member or a very close friend, only because the person who had chosen to take their own life did not want their friends implicated in any legal problems. Because technically, they could be charged for assisting a suicide.
Yeah.
So people would say goodbye, they'd leave, and then the final exit came. But again, it might sound like a horrible thing, but it's also, from everybody I've heard, a very beautiful thing. As you said, you get to leave on your own terms. You have everyone who loves you around you, and you're laughing and joking and telling stories, remembering the past, remembering the good times. And all of your friends, your family, have that one last chance to say goodbye and to say the things they've always wanted to say.
Jade, when you finish, I'd like to add to that.
I'm done. Go ahead.
Okay. My partner never thought about that. That wasn't an option for him. He was going to stick it out till the end because he was at home.
Yeah.
And of course I was there, and the people from hospice were coming by and from the church. You were talking about saying goodbye. Before dementia set in the last maybe couple of weeks, we talked about a lot of things about the past and problems we had, and like we told each other, about things. I am so sorry I said this or did that. And he talked to his family like that too. They would talk about the past, and like one of his sisters, they didn't get along, and he tried to mend things. Because it's like he tripped me out sometimes. It's like he knew it wasn't going to be much longer, and he pretty much told his family and friends goodbye, before things got real bad, and tried to mend bridges and apologize for things, and so people wouldn't be like, well, I wish I had said this or that. He gave everybody a chance. And a lot of people, like you said, at those parties, here was their chance. But a lot of people don't have that chance. And that's such a sad thing. It really is. So, because see, I was with him when his heart gave out. That was not easy. But with someone like that is so open to talk about things, even difficult things, problems in the past or whatever. In a way, it makes the passing, like he was talking about at the parties, the passing, losing someone you love like that, it doesn't hurt quite as much because you don't have that sense of. I know people do it; they may not admit to it, but you have a sense of guilt, like, well, why didn't I do this or why didn't I say that? See, it wasn't like that, with him. He took care of all that before the time came. Anyway, I just wanted to add that to what you were talking about.
Well, thank you, because that does put a personal touch on it. But yeah, that chance, you're right, the chance to mend bridges, the chance to say the things that you never said before but always wanted to say, the chance to apologize for things that happened in the past, and leave on a good note. Yeah.
I think on that note, I'm going to wrap this up.
Yeah. I was going to mention one interesting thing, and I'll just mention it briefly, just as an interesting thing. And it's something worth looking into, and I'll give people the link if the freaking thing comes up. There's actually an association between the Black Plague and HIV. And my computer is working very, very slow, so I can't get to my link right now. So maybe I'll just have to wing it. Here it is. So, well, it turns out that I'll just go ahead and read the one paragraph from this. I'll leave the link later. It said, scientists estimate that the plague killed nearly half the population of Europe in the mid-1300s, but what happened to the other half? NIH researcher Stephen O'Brien's work with AIDS identified a particular genetic mutation, Delta 32, that prevents both HIV and the plague bacteria from entering human cells and causing infection, which geneticists call a positive mutation because it confers survival benefits on those who carry it. So, as O'Brien explained to the PBS Secrets of the Dead, both the AIDS virus and plague bacteria hijack the body's white blood cells. In the case of plague, this spreads the infection to lymph nodes. O'Brien discovered the same positive mutation in about 14% of the living descendants of plague survivors from the English town of Ely. He hypothesized that the mutation spread after the plague because survivors carrying the mutated allele reproduced together. This same mutation explains why some European populations are less susceptible to HIV infection. And there has been some research that showed that, European populations and other populations that survive, that are descended from survivors of the plague, have had less susceptibility to the virus than perhaps, for example, people in sub-Saharan Africa whose ancestors never had the plague. It's an odd little quirk.
That's very interesting.
And I'm done. Yeah.
It's something worth maybe looking at another time. Right, listen, I'm going
Yeah.
to have to wrap this up, guys. I want to thank everybody that's come along to share. And special thanks to Heavy Hauler. Very special thanks to Heavy Hauler. And for Mel for putting together all those links for us. I think the only way to really finish this is to say, everybody, tonight, hug your loved ones even closer than you normally do. Say night-night, everybody.
No, everybody.